Hi everyone, thanks so much for joining us today. We're going to get started at noon Eastern time, so in about 5 minutes. Hi attendees, thanks so much for joining us today. We're going to get started in a couple minutes, so at noon Eastern time. All right, we're going to go ahead and get started. So hi everyone, thank you all so much for joining us today. Welcome to the NOAA Omics Seminar Series. I am your host, Catherine Egan. I sit in NOAA's Office of Ocean Exploration and Research, or NOAA Ocean Exploration. I actually want to take a moment before we get started to introduce our brand new host of the Omics Seminar Series, Nicole Miller. Nicole is our Omics Program Specialist here at NOAA. So Nicole, I don't know if you want to hop on real quick and just say hi.
Hi, everybody. Thank you so much for being here today. I'm very excited to take over this role from Catherine Egan as the new NOAA Omics Seminar Series host. Please keep an eye out for communications from me moving forward, Nicole Miller, and I really look forward to continuing the series and having some really great discussions. Thanks.
Thanks so much, Nicole. Yep, and thank you all for continuing to support the Omics Seminar Series. And that being said, if you have any questions, you could still reach us at our dedicated email address, noaa.omics@noaa.gov. So with that, Omics, which is a suite of tools used to analyze DNA, RNA, proteins, and metabolites, has become a really large priority here at NOAA in the last several years. We established this seminar series in an effort to increase transparency and collaboration and highlight the incredible omics-related research currently underway within and outside of NOAA with our partners. So the seminars from this series are made available on YouTube and posted up on our Omics website. There should be a link to the website in the chat now. As participants, you are muted, but feel free to type questions into the questions for staff box throughout the presentation. At the end, I will read the questions for the presenters to then answer. So with that, we have our presentation today, which is titled Marine Fish eDNA Metabarcoding, Promising Developments and Early Applications, from our two speakers, Mark Stoeckle and Jesse Ausubel. So Mark and Jesse, thanks so much for joining us today. Mark, I'm going to go ahead and pass off screen sharing permissions for you so you can get set up. And while that is happening, I will go ahead and introduce you to our audience. So Mark Stoeckle is the Senior Research Associate in the Program for the Human Environment at the Rockefeller University. So Mark, thanks so much, and you can go ahead and begin.
Thank you, Nicole. Thank you, Catherine. My colleague Jesse Ausubel and I are going to talk about marine fish eDNA metabarcoding, promising developments and early applications. First, I want to thank our contributors at Rockefeller, Zachary Charlotte Powers, Michael Kukin at University of New Hampshire, Jason Adolph and Keith Dunton at Monmouth University, and Greg Hinks and Stacy Van Morter at the New Jersey Department of Environmental Protection. We thank NOAA for supporting this work. The sources for what I'm going to present are in 3 recent publications and from work in progress. I'm going to say 3 main things. First, eDNA abundance matters. It's relevant to detection, quantification, field design, and laboratory protocols. Second, that adding an internal standard to metabarcoding PCRs quantifies eDNA. It allows you to convert relative sequence reads into absolute eDNA copies. And third, that current marine fish metabarcoding protocols are ready for wider use. They provide a reasonably accurate index of fish abundance, especially for more abundant species. And Jesse's going to talk about eDNA metabarcoding application in ecosystem-based management. As background, I think it's helpful and important to think about the distribution of marine fish abundance. Like other animals, marine fish abundance typically follows a hollow curve. This is data from the New Jersey Trawl Survey, monthly catch weights, January 2020. And the graph on the left, we have species weights, and species, each column is a species. Species are ranked by decreasing weight. 40 species were captured that month. A few species are abundant and the rest are increasingly rare. And this is the same data shown in a log scale. A few species are abundant and the rest are increasingly rare. And this abundance ranges over 5 orders of magnitude. And given this very wide range, I think order of magnitude accuracy is a reasonable starting goal for eDNA versus fish abundance. If we can do that, that's already a big step forward. And second, that detection and quantification of uncommon species, these ones over here on the right of the graph, it's probably going to be challenging by any technology, including by eDNA. we already know that metabarcoding reports fish abundance, at least under some circumstances. In these circumstances, it must mean that metabarcoding reads are at least roughly proportional to eDNA concentration, and eDNA concentration is at least roughly proportional to fish abundance. For example, we can see that metabarcoding reports changes in species abundance by season. This is data from the New Jersey Trawl Survey starting 2019, January, June, August, November, and January. And here, each column is a separate trawl during that month. And the columns are colored according to whether these represent a set of cold water species, spiny dogfish, little winter skate, and alosa herrings, or warm water species, northern sea robin, clear-nosed skate, and bullnose ray. And you can see a dramatic change in what species are abundant in January as compared to June and August, and then we go back to the colder water species in November. This shows the temperature. January it's cold, warmer in June and August. November starts to get cold, and January cold again. We see the same switch in abundance if we do eDNA. So this is during this trawl, we collected water samples, analyzed those, and again, we're showing here percent of reads that are total reads for each sample that are due to the cold water species or the warm water species. And we see the same switch in abundance, including that in November, when it's still a little bit warm, we still have some of the warm water species. We see that in the trawl, and we see that in the eDNA. You may be wondering, why are these columns lower on average than these columns? I think the main reason is that the trawl is capturing fish that are right on the bottom. The eDNA is picking up fish that are throughout the water column. So we've been testing an internal standard added to metabarcoding PCRs as a way of quantifying marine bony fish eDNA. Our main metabarcoding target is 110 base pair segment of vertebrate 12S ribosomal RNA gene, so-called Riaz segment. This is immediately adjacent to the MyFish segment, another metabarcoding target for vertebrates. And we did some comparisons of these two. For a DNA standard, we used an amplicon prepared from ostrich or emu eDNA 750 base pair amplicon that contains the metabarcoding regions and has identical primer binding sites for the Riaz primers, forward and reverse primers. So our main question has been, are reads directly proportional to copies? If that's so, then replicates with different amounts of standard should indicate the same number of copies of fish eDNA. So this is how we tested this. We made a spiked metabarcoding replicates with a DNA standard. So a set of replicates containing the same amount of fish DNA, same proportion of a sample, and different amounts of a standard, sorry. We then prepared libraries from these, individual libraries, one replicate, one library, and looked at how much of the reads were due to ostrich or standard, due to fish, or due to other vertebrates. And then we can take the proportions between the standard and the fish and calculate how many copies of fish DNA it seems that are in there. And if we take the 600 copy library, the 60 copy library, we get about the same answer as to how many copies of fish DNA there are. So this one experiment, it says that for this, reads look, roughly speaking, directly proportional to copies. I should say this approach, the spiking approach, was first used by Ushio and colleagues in 2018. We can also take this number that we get for fish copies in the sample and look at individual species. And so this looks at copies per species in the 5 replicates. Each species is a different color, and lines connect detections in successive replicates. You can see for high copy number species, this is a log scale, this is 1,000 copies in the eDNA aliquot we analyzed. The results are really very reproducible. When you get to lower calculated copy number, there's more variation. And at the lowest range, there are species that show up in one sample and not in another. We tested this protocol on more samples, we got the same result. This is 18 different DNA samples that we analyze with a 6,000 copy standard or 600. And when we calculate how much fish DNA is in there, the 600 copy standard gives us the same answer as the 6,000. We also did that with 600 versus 60 copies of standard. Here we use some of these, we used emu DNA, that's in gray, versus ostrich is in black. And again, a linear relationship over a wide range, and same thing with 60 or 6 copies of standard. So we have a linear relationship between copies and reads over 6,000, a 1,000-fold range of standard, 6,000 to 6, and about a 1,000-fold range of fish eDNA. This is about 5 copies of fish DNA, and this is about 5,000. If we look at the difference between these two single replicates, the average arithmetic difference is about 1.4-fold, and the maximum is 2.3-fold. So this looks good in terms of converting reads per sample to eDNA copies per sample over this range. And this approach, as I mentioned, was first used by Ushio and colleagues in 2018. They call this Q, MyFish, Q for quantitative, MyFish because those are the primers that they use, the metabarcoding primers. It's also been applied more recently by another group, and you see a reasonable linear relationship between qPCR copies and the Q MyFish copies. This spike-in approach has also been used in microbiology. There's a linear relationship here over 7 orders of magnitude. Sorry. This approach identifies a threshold for reproducible amplification, which in our samples is around 10 eDNA copies per reaction. We looked at this by taking these 5 replicate samples. We had 8 sets of 5 replicates. We calculated the copy number of each species and then how many replicates had appeared. And then now we've ranked all of these detections by the species, calculated species copy number. You can see above 10 copies per reaction, 5 of 5 replicates are positive. And as the copy number goes down, the input copy into the reaction, we get more and more dropouts. So this provides an intuitive explanation just for the phenomenon of dropouts. One other important thing is in our samples, most of the detections are below this 10 copy threshold. So 80% of the detections are below this threshold for 100% detection with single replicate. We tested this on more water samples. We got the same threshold. This is 16 water samples from New Jersey survey of Raritan Bay last year. 2 replicates per sample, either 300 copies of standard or 30. And again, we have a close relationship to identity here between what we calculate as this is now copies per species, not total fish DNA, that we get with the 30 copy standard. It's the same as we get with the 300 copy standard. And here again, below 10 copies, 10 to the 1 is 10, there's more noise in this, more variation, and there are dropouts. These are, you could either call these singletons or dropouts. These are detections that were only in the 300 copy standard or in the 30 copy standard replicate, and essentially all of these are below 10 calculated copies. Are these results consistent with qPCR studies in general for marine fish? And I'd say yes, among the relatively few quantitative marine field studies we've found so far. This is data from Ollie Shelton's and others' recent work, the very large qPCR study of Pacific hake. They looked at number of samples and the eDNA copies per sample by qPCR for Pacific hake. This is at the 100-meter depth, and you can see their detections range from about 10 to about 10,000. And this is compiling the data that I just showed you from the Raritan Bay samples, 16 samples. And if we convert these into copies per liter, they range from about 10 to about 10,000. So this looks promising. This approach also can overcome distortion due to amplification of non-fish eDNA. For example, this is looking at water samples from the East River, either 1 liter or 2.5 liters, filtered, suspended the DNA in the same volume, and 5 replicates, again here, same amount of DNA in each one, different amounts of the ostrich standard. And although we started over here with more water, so presumably should be more fish DNA, actually the fish reads are fewer, and the non-fish reads are greater in this set of samples. But if we take the standards and then use those to correct, calculate the actual number of fish copies, we get what you would expect. So a lot fewer fish copies from 1 liter than we get from 2.5 liters. Okay, that looks very good, but what about primer bias? This is an excellent talk just preceding in this series from Zach Gold towards quantitative metabarcoding, which he detailed circumstances with very significant primer bias and presented ways of compensating for that. Well, focusing just on our system, which is looking for this moment just at marine bony fish with this particular either MyFish or Riaz primers, we reasoned that if there was significant primer bias, we would expect reads per species to differ between primer sets. So this is taking a set of samples. This is reads per species analyzed with Riaz primers or with the MyFish primers. And over most of the range, you get pretty much the same result. The Riaz primers are giving the same. So at least in this experiment, it doesn't look like there's a lot of primer. There are not hints of primer bias here. There is more noise at the lower end, below about 1,000 reads here. But we've done this with other samples, and our conclusion is that primer bias is probably not a major limitation in these experiments. That's specifically talking about marine bony fish with MyFish or Riaz 12S primers. This is looking at that data a little more detailed. This is the graph I just showed on the previous page. I forgot to mention that the dropouts or pickups were the number of them was the same. So 6 species only detected with MyFish primers and 5 species only detected with Riaz primers. If we look at reads above 1,000, this is really a very tight correlation. And then below 1,000 reads, which we think is from low copy DNA, there's a lot of variation. We think this is consistent with stochastic effects of rare eDNA rather than primer bias. This picture up here is just to remind me that it doesn't have to be, assay doesn't have to be perfect. Just as some fish escape the net, we may have some degree of primer bias with our assay. As long as it's reproducible, we can work with that. There's also other indirect evidence that primer bias in this setting is not significant. I gave you one example at the beginning, that the eDNA is showing you a similar thing that the trawl is showing you. This is also from that same study, looking at trawl weight. Each point here is a single species in a single month. And this over here is percent reads. And there's a good correlation. This is R squared 0.71 over multiple orders of magnitude. We can actually improve this correlation if we do allometric scaling of the weights, go from 0.71 to 0.77. And this approach is detailed in a recent publication by Matthew Yates. And I think this is going to turn out to be very important for relating eDNA abundance to fish, that in a certain way that eDNA is probably more proportional to external, internal fish surface area than it is simply to biomass. But at least in the system we're working with, we think primer bias is not a major factor. We do see it with cartilaginous versus bony fish. So this is looking at but here it's both predictable and solvable. This is looking at the Riaz primer binding site, forward primer, in a set of regional bony fish and a set of regional cartilaginous fish, essentially no substitutions among bony fish, but all of the cartilaginous species have 2 substitutions, in particular T at the 3' end of the forward primer. So we tried modifying the forward primer. Instead of having a C at the end, we put a T. Actually, this primer works pretty well for bony. This is just bony fish. This is reads per species, again, on a log scale. The blue line is what it would be if the 2 primer sets were giving you identical results. So it is a step down, about half an order of magnitude or so, but it's consistent over the range of abundance here. There's a very dramatic difference using this primer versus the bony fish primer for cartilaginous fish. It's about 2 or 3 orders of magnitude better at amplifying cartilaginous fish than the bony fish primer is. The fact that this isn't symmetrical is predictable also, because here you have a TG base pair mismatch at the 3' end, which is relatively okay. And here you have a CA mismatch at the 3' end, which is not favored. There are limits to the internal standard. A very high amount, maybe you can imagine, will suppress fish reads and detection. This again is looking at 4 water samples. We have a set of 6 replicates here, same amount of DNA in each one, different amounts of ostrich standard, and we're showing percent of the total reads from the MySeq run. If you start out with 60 million copies of standard, 600,000, 6,000, not until you get down to 6,000 you start to see any fish reads. So this is a lot of standard, and you might get the same effect if you had a fish DNA in your sample that was also very abundant. Surprisingly, the species detections, even though the number of reads here is quite small, the species detections, that's the black line, it's pretty good, even though we've suppressed the read counts. So what have we learned so far? That metabarcoding spike in adds standard adds value at no additional cost, at least if you're already doing separate replicates. It identifies limits and potential improvements to species detection and quantification. The current metabarcoding protocols with a spike-in standard report absolute eDNA abundance with reasonable accuracy. This is for marine bony fish, particularly for more abundant eDNA. And conversely, that, as many people have emphasized, that species and eDNA rarity is the main constraint. Why do we get dropouts? Part of that is math. Imagine if we had a 100 microliter DNA sample, some of the eDNA in there is going to be rare. If we take an aliquot of that, 5 microliters for our PCR, some of the time we're going to be picking up those rare eDNAs, and sometimes we're not. And there are other rarer eDNAs out there floating around in the ocean that when we collected the liter of water and concentrated the DNA, we didn't pick them up at all. If with our protocol, using a 20th of the DNA from 1 liter of water, we can translate that into eDNA copies per liter. So about 200 copies per liter with this protocol, single amplification, we get very reproducible amplification. There's a range that's variable, and there's a range that their DNA is either they're rare in your sample or they're not even in your sample, that your chance of detecting them with a random sample is going to be very small. Looking ahead, we'd encourage use of a spike-in standard in metabarcoding PCRs. As many people have emphasized, less abundant species may need more water or more DNA or both. Deeper sequencing alone is unlikely to be helpful in this setting. It's unlikely to be helpful because there just isn't that much fish DNA in a liter of ocean water, at least where we're collecting it, so that the range between the most abundant fish and the least abundant is probably 1,000-fold. And if you're doing 100,000 reads with MySeq, you should be able to capture that. Certainly more work is needed regarding primer bias, allometric scaling. There's a nice online tool in Matthew Yates' recent publication. We need standards for other taxa. What I showed you was just bony fish, but we should be able to do it for cartilaginous fish and other taxa. And also to look at water samples from other environments, which may have higher levels of fish DNA than what we're seeing regionally. And again, we'd say that current 12S metabarcoding protocols with a standard, they're ready for wider use in assessing marine fish abundance, especially for more abundant species. So I'll stop there and turn this over to Jesse. Thank you.
Thanks so much, Mark. And yes, for our audience, please feel free to put questions at any point during either presentation into the questions box, and we'll get to them at the end. So thanks, Mark. So Jesse, I'm going to go ahead and move screen sharing permissions over to you. And while Jesse is getting set up, I will quickly introduce him. So Jesse Ausubel directs the Rockefeller University's Program for the Human Environment. So Jesse, thanks so much for presenting, and I'll now pass it off to you.
Okay, thanks, Catherine and Nicole. Can you see the next slide now?
Yes, looks good.
Okay, good. Okay, well, this is a NOAA seminar, and we want to relate what we've been learning very directly to the challenges that NOAA itself faces. And some of the people on the call will certainly think back to 2016, when NIFS implemented for the first time a policy of ecosystem-based fisheries management. The document is here. Many of you know Mike Sissenwein, Mike Fogarty, others who've advocated for this over the years. The policy was adopted in 2016. It was renewed in 2018. It will be reviewed again in October of this year. But as we know, while many people say good words about ecosystem-based management, it's been hard to implement, not only in the context of fisheries, but it could be marine sanctuaries, coral reefs, other situations as well. And not only for NOAA, but other federal and state agencies as well. And one of the reasons has been that to do ecosystem-based management, we need more frequent, comprehensive, multi-species monitoring. And what I'd like to now, let's see, hold on. What I'd like to now argue is that based on the work that Mark presented, we are ready to do that. And in fact, there's an urgency about doing it. Here you see a map of offshore wind development from the 2022 Mid-Atlantic State of the Ecosystem report prepared by NOAA. And there's a lot of development going on and proposed. We've been reading a lot about beached humpback whales in recent weeks. There's a lot of interest in what's going on over these large areas. The assumptions that people have been making in fisheries or other areas, we've been focused on single species, one species at a time, whether it's humpback whales or cod or lobsters. And part of the problem there is that it assumes a stationary production function over, not from year to year, but over the long term, over decades and more. But we know that if we build wind farms, if we build ports, if we lay seafloor cables, if we have oil and gas operations or spills, we know that things change. So the stationarity is actually a poor assumption, and all of the species, of course, exist within complex dynamic ecosystems. The humpback whales care about the menhaden, the cod may care about the lobster, and vice versa. And there are multiple system drivers now, as the very good State of the Ecosystem report describes. There are changes in climate, there are changes in chemical pollution, there are changes in soundscapes. So lots is happening at once. So the assumption of stationarity is probably the least defensible one. So along with the dynamics of the ecosystem itself, the ecosystems on their own might shift from one regime to another, but particularly with the increasing drivers and the strength of the drivers, there's more and more concern that what may happen is that we'll shift from one regime to another. And here you see a figure on the left with, let's say, humpback whales. On the right, the whales are gone, more squid. So, and this can be in the benthos, it can be in the water column. But how do we go about knowing whether there's been a regime shift? Trawls are hard to conduct, expensive to conduct. Here you see the history of the for the Mid-Atlantic, the Northeast Fisheries Science Center bottom trawl data for 28 different fish species for the Mid-Atlantic, showing five orders of magnitude, to go back to Mark's orders of magnitude, let's say from 1, 10, 100, 1,000, 1,000, 10,000, but here just say good condition, above average, neutral, below average, poor condition. Very general descriptions of the stock. And of course, a survey like this, using the Albatross, the Bigelow, or new vessels, it's hard, it's expensive, a lot of diesel fuel, and in fact, it's missed sometimes. Here, as you see, the 2017 white stripe, the survey wasn't conducted due to delays, and of course 2020, because of COVID, it wasn't conducted. And some years there are no data on particular species, on spots or bullnose ray or offshore hake or others. So it's been a tremendous challenge, not only for NOAA, but for counterpart agencies in Japan or in UK or Australia or New Zealand, to have enough data year in, year out, more than once a year on, let's say, in the case of the fishes, 28 different species of fishes at a time. Mark began by showing these figures, the New Jersey Trawl Survey, and what we would say now is that these distributions, either on the linear scale or the log scale, call it a rank size distribution, say a rank weight distribution, but classically in statistics, one would call this a rank size distribution, that this distribution, in fact, defines an ecosystem, or it characterizes an ecosystem empirically. These actual data, the composition, the order of the species from 1 to 40, the membership, which 40 are here, what order they come in, the level, how high on the scales of abundance each species is, and the slope, the steepness of the slope. We would say these features define an ecosystem. And if there's a major change in the order or the slope, we would say that constitutes a regime shift. And here it's fishes, but of course one could include more taxa, cetaceans, pinnipeds, invertebrates if one wished, but it may well be that using the fishes is highly representative. And of course, in single species management, in a sense, you're paying attention only to one column of the 40. But if you really want to do ecosystem-based management, you want to look at the entirety. Well, the good news is, as Mark showed, the eDNA is doing the job. On the left, on top, you see the trawl biomass, and on the lower part of the left, you see the species ranked by decreasing weight, or reads, copies from the eDNA, with actually 71 species rather than 67. And correlated very nicely. On the right, you see that the eDNA reads correlate very nicely with the trawl weight reads, and as Mark showed, actually even a little bit better with the allometry. So the great news is that we think the abundance data, the ability to create the profile, the ecosystem profile, it works, it's good enough. And the standard, as Mark showed, the standard lifts the quality even higher. Using survey vessels like the kinds that NOAA uses, but it could be in other countries as well, costs between about $15,000 and $50,000 a day. By the time one pays for the crew, the vessel, the diesel fuel, we won't even speak about the carbon footprint. eDNA collection, let's say by a drone or in a very small vessel, can cost 10% of this. And you can do it more frequently. It's not limited to once a year. You don't have to skip a year if there's COVID. And you could do it more densely if you want to do it as well. And it introduces the possibility of comparability across all samples and zones. Here you see for New Jersey also the surf zone, very hard to sample. I mean, a vessel can't get into the surf zone, or in the estuary, in estuaries where again it's very hard to do trawls. Very shallow draft vessels are needed, and the eDNA gives an identical method that can be used across all the zones, including those that are hard to sample. And with the kind of spiking that Mark talked about, one can standardize this for all the, I'll say all the ecosystems. So I want to conclude there. Thank everybody again. And we think the abundance news is really important, whether it's for characterizing marine sanctuaries, for emergency situations like oil spills, and obviously for fisheries. We think the eDNA abundance data methods, we think it's ready for prime time, and we think it can enable in the very near future, maybe even 2023, somebody has to go first. We think one can really start to implement ecosystem-based management, move beyond the single species management this way, characterizing the ecosystems, looking for regime shifts, and managing many species at once, the metabarcoding approach. So thanks again to everyone for the contributions, the New Jersey team, NOAA itself, and above all, Mark, who's led the lab and analytic work on this. Thank you.
Thanks so much, Jesse. Thank you both, Jesse and Mark. This was a really interesting seminar. Hear about the science and then hear about the management applications thereafter. We have a question that came in, but real quick, folks, please feel free to type your questions into the questions for staff box. We have plenty of time to answer and also some time here for further discussion as well. So our first question, I think, Mark, this one is directed to you, but I'll see if you'd like to answer or if you'd like to pass off to Jesse. This one's from Nancy. I had a question regarding your study on the fish DNA. How do the choice of primers and the length of the target DNA sequence impact primer bias in quantitative metabarcoding?
I'll give you the answer that I know, which is, as I said, we've really focused on using this one particular 12S primer pair, the Riaz primers, and comparing it to another short region, the myfish region. So we haven't attempted to test the issues in general about target length or in specific with other primer sets. So I can't, we just don't have data on that. It's a good question.
Thanks, Mark. We have another question that came in for you from Andrew. They say, excellent presentation, thank you. I would be keen to hear your thoughts on incorporation of standards into eDNA metabarcoding approaches that target different species through the incorporation of taxon-specific primer sets. Is the incorporation of multiple standards necessary to deal with this added complexity?
The approach, that's an excellent question. The approach that we've been taking is to make the experiments absolutely as simple as we possibly could, only to answer one question at a time, which is why we started out doing separate replicates for the two different standards, comparing 600 to 60 copies, for instance. It would make sense, and what other people have done is to have multiple standards in a single reaction. We just haven't done that. Someone needs to, and maybe people are working on that. So I don't know, and if you then start doing multiplex amplification, which I think is the future, you kind of have to go through systematically and build up and see that it's giving you the same answer you're getting with a single amplification. So we haven't tried to do it, but I think it's important to develop that.
Thanks, Mark. This next one might also be for you, but once again, we'll see if you want to punt it off, Jesse. This one's from Daniel. I'm curious about the logistics of the eDNA collections. Are the samples being collected from the surface, mid-water column, benthos? Are there differences in relative eDNA abundance based on the behavior of a particular species?
Our samples are, for the trawl study, they were collected at surface, one at the surface and one at the depth before about a quarter of the trawls. The deepest water that the trawl goes is about 100 meters. So that's as deep as we've gone. We haven't, questions about, and we've done a lot of surface or near surface samples in multiple environments. So how does the overall abundance of eDNA, differ? I think just that's something we need to start looking at.
Thanks, Mark. Okay, this one's not so much a question, but something for both of our speakers and also our audience to ponder. This one's actually from Craig McLean. Thanks, Craig, for being here. He asks or says, curious from the nymphs or fisheries colleagues as to how long would parallel comparisons need to be conducted in order to shift reliance on eDNA and use trawls as a cross-check. So Jesse or Mark, I don't know if either of you want to maybe address that as well.
Jesse, maybe you can comment?
Yeah, thanks, Craig. First, we would say it's not an either or. Certainly one wants to maintain the trawls for a number of reasons. I think but as you say, but one wants to have some overlap. And there'll need to be overlap also for cephalopods or for other invertebrates or mollusks. They'll be learning for different taxonomic groups. So in that sense, it's going to be a few years of this kind of I'll say of overlap. If it turns out that each group, let's say we try cephalopods next, if it turns out that the first 3 or 4 groups that are done all are as good as the fishes, then our confidence will grow rapidly. If it turns out that some of the groups don't do as well, then and things are really very taxon specific, then obviously we need to think more. It could also be that there are differences in terms of other features of the water, that in cooler water, more northerly areas, the things are more consistent. So than in some tropical areas, Hawaii may have some somewhat different issues. So there'll need to be what Mark and company did for the New Jersey Trawl Initiative. That's going to need to be done quite a few times. That said, I think the incremental or marginal cost of the eDNA collection and analysis is low. And so I think what we might look to is a world, at least let's say for the next 10 or 15 years, in which we try to continue doing a lot of what we've been doing in terms of monitoring, whether it's in marine sanctuaries or by the Fisheries Service, but that we, to the extent we can afford it, we start doing the eDNA surveys also, but maybe quite frequently, maybe 4 times a year or 6 times a year. And it may turn out that, again, that the incremental or marginal cost of doing that, once there are personnel who say the people in the training may be the expensive part or the slow part. The collecting, the physical, the logistics, and the sequencing costs, all of that is really very low. I'd say it's having more people like Mark and Jason and Keith at Monmouth who can do the analytics, or I'll say Ryan Kelly and Kelly Godwin in Seattle. So we need to grow a generation of people who can do the analytics as well. But I think we should. It works so well already that it seems to me a bit crazy not to do a lot more of the overlap studies starting tomorrow. Thanks.
Thank you very much, Jesse. And I really encourage any of our NOAA fisheries colleagues who are listening in, if you'd like to make any comments, you can once again type it in the questions box and we'll circle back around to it. Okay, our next question is from Trey. They ask, how are species-specific estimates of abundance obtained from multicellular organisms? It seems straightforward for unicellular organisms, but unclear on how this would apply to an organism that can shed thousands of cells frequently. I think maybe this one goes to you, Mark.
Yes, if I understand the question, in best circumstances, we're using the trawl. How do we count fish now? I mean, we use trawl surveys or visual surveys, but in our region it's mainly trawls. So that's our anchor for assessing fish abundance. That's the traditional method of doing that. And then we're taking the eDNA levels we get and seeing how they correlate. So you have to count the fish.
Thanks, Mark. Our next question is from Pooja, and they ask, how can quantitative metabarcoding be used to estimate the abundance of a species in a sample?
That's the data. We have one set of data here. Hopefully, this use will be expanded. The main observation is that the metabarcoding reads are directly proportional to the copies of eDNA in your sample. So if you add a standard to your sample and you know how many copies that has, then you can calculate how many copies of Atlantic herring DNA are in that sample, or how many copies of summer flounder in that sample. So that's, and what we need more of is the thing that Jesse is talking about, where there are multiple comparisons from eDNA data to a traditional survey technique.
If I might add, there is, of course, a craft in going from I'll say the raw abundance information, whether it's from a trawl or from the eDNA, to an assessment of ecological condition. And in the figure from the NOAA 2020 trawl, you see that obviously there's a lot of argument among experts within the groups and sometimes in the courtroom about how to go from one estimate to another. That's the basic quantitative information to the quintiles of condition. And here I would come back again to the idea that the metabarcoding would allow a more objective way of estimating or of characterizing abundance. Again, we can use the rank size distribution or the rank weight distribution, and as we build up time series of those, which I think we have the potential to do because of the lower cost of the eDNA collection and analysis, then that you could imagine those charts, those figures, those data sets themselves becoming the way we talk about ecosystem condition, and sort of moving away from, again, from this trying one by one for hake or for lobsters, trying to assess condition, but rather move in the direction of using this, the membership in the group, the 1 through 71, their order, the height and the slope, and maybe 15 or 20 years from now, that will be the main way we think about reporting abundance, and you could say condition of marine life and collections of marine life in ecosystems. Thanks.
Great, thank you both very much. We have a couple other questions coming in. This next one is also from Trey. They ask, have comparisons been made in estimating relative abundance in areas other than sounds and rivers? I assume modeling is required to account for oceanographic conditions, as well as an understanding of DNA degradation rates and shedding rates of target species.
I'll make some comments. The ocean's obviously a complicated place. We haven't done the modeling end of it, but people are certainly doing that. Elizabeth Allen has done a lot of work, sort of particle tracking, and there are a number of mesocosm experiments looking at shedding rates for fish and different species, different temperatures. The simplest anchor to me is to take a traditional measure. I think this is a personal view. I think it's going to be very hard from first principles, if you know shedding rate, ocean temperature, ocean current, to predict what's going to happen to the eDNA. You can work backwards from abundance, though. So, but yes, people are working on that.
If I might just add, the same contextual and complementary information is needed to make trawl data useful. There are always the concerns with trawl data that, let's say, because of an early or a late spring, that the what was found in the nets wasn't representative. If it had been 2 weeks earlier or 2 weeks later, it would have been different. Or that this year the anchovies were simply deeper because of different thermocline. And so when the contextual information about oceanography and so forth is important, whatever method one is using. Finally, we would look forward to a world in another 10 or 20, 25 years in which we're using a combination of methods, also acoustics, where we can compensate for the weaknesses of each method by integrating information from several. Thanks.
Great, thank you both. Okay, we only have a few more minutes left for questions, so I'll try and power through some of these. So this next one is actually a comment from Rich McBride. So Rich works for NOAA's Northeast Fisheries Science Center. He says, hi Mark and Jesse, thanks for the updates. Our updates from NOAA's Northeast Fisheries Science Center is that Yuan Liu has sampled eDNA across continental shelf in 3 seasons now, spring, summer, and fall. We presented the preliminary results at the recent Flatfish Biology Conference, and he also sent a link to those abstracts, which I can pass along to you too. I don't know if either of you wanted to make a comment, or we can move on to the next question.
I'd say, well, hi to Rich, and Yuan is doing terrific work, and that's great to hear. And that kind of 3-season work, it's exactly what's needed. That's again, as we've all been saying, and as Craig McLean's question, I think NOAA and other agencies, Bureau of Ocean Energy Management, we need a kind of, I'll say a national plan. I don't mean a big bureaucratic plan, but we need strategies, plans, coordination among the different groups interested in these kinds of things, so that among us, over the next 3, 5, 10 years, we cover the waterfront, so to say, and the kinds of experiments and surveys to answer the outstanding questions. Thanks.
Thanks, Jesse. And just a quick plug that Yuan Liu, who's a researcher with Northeast Fisheries Science Center, actually gave a presentation on some of her own research for the seminar series. You can go look at it up on our Omics website. Okay, we have one last question, unless any others come in the last 5 minutes. This one is from Rebecca, and I think, Mark, this maybe is a question for you. Can you determine the state of health of organisms from the eDNA collected?
That would be good. From what we're doing, no. We're looking at DNA. DNA could be shed by a living organism. It could be shed by a dead organism. There are many people working on eRNA, which does have the potential to look at species metabolism and potentially species health. So, the technologies are potentially out there, but I don't have any specific information on that.
I would add that the capacity to look at 71 species at once does introduce the possibility to look at effects on clusters. So, if one sees again that several species that have, let's say, similar roles in an ecosystem drop out, or that their biomass is way down, then that could certainly be a clue about a problem. Again, in contrast to only looking at one species at a time. So while it's not the same as actually having an organism and looking to see whether it has a virus or a tumor, or effects of, let's say, some kind of chemical pollution, there still may be ways, again, to use the metadata to learn something about at least health of clusters of taxa. Thanks.
Great, thank you both so much. That actually concludes the question portion of our seminar. We didn't have any others that came in, so I'm going to go ahead and wrap this up. So Jesse and Mark, thank you both so much for taking the time to present today for the seminar series, and thanks so much for all of our attendees for listening in. The NOAA Omics Seminar Series generally takes place on the third Wednesday of every month at 12 p.m. Eastern Time. However, our next seminar is coming up a lot quicker than that. So our next seminar is going to be in 2 weeks. It's on March 15th at 12 p.m. Eastern. It's titled Microorganisms and Metabolites as Diagnostic Indicators for Coral Reefs. Our presenters are Drs. Amy April and Elizabeth Kujawinski from Woods Hole. So registration is up on the Omics website, and thank you all again for joining today.